about the effects of maternal exercise on offspring. These actually are my offspring. And so what does that have to do with IBEC? So let me get the pointer here. So if you look here on the left, this is my daughter. Her name is Sydney. And why is she named Sydney? Well, in 1997, I was invited to speak at IBEC, which was in Sydney, Australia. Some of you know my husband. He's a little bit crazy. And he said he got turned upside down by going to this trip, and he proposed to me the last night of the IBEC meeting. And then a whole bunch of us, many of you who are here, went out and celebrated. So I have a very strong, warm memory of my first talk at IBEC, and Sydney was named after that trip, obviously. So today I'm going to talk about work on maternal exercise, and we all know that there are many negative effects of obesity on health. This is in contrast to exercise that has so many beneficial effects of exercise on health. This is a huge problem because worldwide there are very high rates of obesity. Over 2 billion people in the world are estimated to be obese. And so why does this happen? Well, we know that part of it is genetics, part of it is lack of exercise, and part of it is diet. But what we think is also important is the fact that there are very high rates of obesity in women of childbearing age. And in fact, the CDC reports that 36.5% of women in the so-called childbearing age are obese with a BMI of greater than 30. So why is this such a big problem, and could this contribute to this epidemic of obesity? And we know that if a mother is obese, if a woman is obese while pregnant, her child has an increased rate of obesity, can have an increased rate of obesity, but that can even carry over then into when she becomes an adult, the child becomes an adult, and there's an increased risk for obesity and type 2 diabetes in adulthood. But the problem doesn't end there, because now there's evidence that then when this woman has a child and that woman becomes an adult, there's also an increased risk for obesity. So if the mother is obese during pregnancy, there is increased risk for offspring from subsequent generations to develop obesity and type 2 diabetes. And we think that this is part of the mechanism that's fueling this worldwide epidemic of type 2 diabetes. So I remember many years ago now sitting in a lecture, hearing about this and thinking, well, there must be a way that we can try to overcome this. And as an exercise physiologist, when I asked this question to myself, I thought, well, maybe exercise can prevent this transmission of disease. And so we would love to do these studies looking at the effects of maternal exercise in adulthood in humans, but those studies could take 40, 50, 60 years. So instead, to address these questions, we've used mouse models in our laboratory. And so we can have the mothers or the dams perform voluntary wheel exercise while she's pregnant, and then look at the offspring and also look at the grand offspring. And the terminology that's used for this is that the mother is considered the FO, and the F1 is the first generation, the F2 is the second generation. And the hypothesis when we started this work was that increased physical activity of mothers will improve the metabolic phenotype of offspring. So today I'm going to summarize the first work that we've done in this very quickly with just a lot of cartoons and just a little bit of data. I'm going to talk about the effects of maternal exercise on offspring metabolic health, and then I'm going to get into the mechanism of an epigenetic regulation, the identification. Then I'll talk about the identification of a placental protein that we think is very important in these responses, and then as Dave showed on the slide, I'm going to take a couple minutes to talk about the effects of grand maternal exercise on the F2 offspring metabolic health. So first, the effects of maternal exercise on offspring health. So this work was spearheaded by Kristen Stanford, who was a postdoc in my lab. You all know Kristen now. She's speaking this afternoon. She's an associate professor at Ohio State. And Kristen just did an amazing job getting these projects going in the lab and continues to work in this area in her own lab. So the first question we really wanted to understand was, what is the effect of the maternal exercise? And we focus on glucose tolerance and insulin concentrations. And knowing these bad effects of obesity, we also wanted to see whether maternal exercise could prevent the detrimental effects of a high-fat diet on offspring metabolic health. So this is the experimental design that we use for these types of studies. Typically, we have one group of females that's chow-fed, and then they're sedentary throughout a period of preconception, during breeding, and they're sedentary during the gestation. Or we have mice, females that are trained for 2 weeks prior. They're removed from the wheel cages, so the males don't have the effect of exercise. They're bred for 3 days, and then put back into the wheel cages where they voluntarily exercise throughout gestation. We also typically also do 2 more groups where the females are fed a high-fat diet both before and during the training period. So do the animals exercise when they're pregnant? Yes, they do. They typically average about 2 to 3 kilometers a day. During pregnancy, it falls off, but on average, it is 2 to 3 kilometers a day, and they have a typical training response that you would see in a non-pregnant female. Okay. Slide is not advancing. Okay. Okay, and also what's important to point out is that the male breeders were sedentary and chow-fed. So the treatment in these studies were only of the females. So again, here we go, only the mother was treated. Keep that in mind. The offspring was not treated. The offspring were sedentary and chow-fed. And we looked at the offspring metabolic health, both from youth up to middle age of about 52 weeks. And I'm going to show you data on glucose tolerance as measured by area under the curve. So the lower the value when we're looking at the area is a better metabolic response. So here are the data. These are the data from when the mom was chow-fed, either sedentary or trained, the trained shown in the black bar. And what you can see is this is the male offspring. We see essentially the same data in female offspring. But what you can see is that as the animal ages, there is a little bit of a worsening in glucose tolerance, shown here. But if the mother had exercised, the offspring's glucose tolerance is significantly improved at middle age. So really quite striking results. Now, if we look at the animals that were high-fat fed, if we look at the offspring from the moms that were high-fat fed, you can see in the red here, if the mom ate a high-fat diet and was sedentary, the glucose tolerance is worse compared to chow-fed, and this gets worse as the animal ages. But remarkably, if the mom had also been in the wheel cages and exercised, this bad effect of the high-fat diet on the offspring is completely reversed if the mom had also exercised trained. And there was no difference in food intake or activity levels in the offspring, so that is not the mechanism. Another really striking finding is looking at insulin concentrations. This is in the 52-week male, and you can see if the mom trained, insulin concentrations were significantly lower. If the mom ate a high-fat diet, insulin concentrations in these males, when they're the offspring at 52 weeks of age, is significantly increased. And again, there's this effect of training, if the mom had trained, to lower insulin concentrations. So these data really, I think, show quite clearly that maternal exercise before and during pregnancy improves the metabolic health of male and female offspring and prevents the deleterious effects of high-fat feeding. So we've gone on, and I'm going to tell you some additional data, but we've seen this consistently in multiple studies and cohorts that we've done, and it's also important to point out that other groups also see these beneficial effects of maternal exercise on health. This includes work from Kevin Pearson and Zanyan and Mindu, some of the papers shown here. So different models and slightly different experiments, but generally the same sort of findings. So we continued on in this work, and one of the important questions we wanted to understand is what's the tissue or tissues responsible for the improved glucose tolerance in the offspring of the mothers that exercised. And while we've looked at multiple tissues, what I'm going to focus on today and what we think is really important is adaptations to the offspring liver. And this then gets into epigenetic regulation of the offspring liver. Oh, and I should mention that Joji Kusayama, some of the work I'm going to show you is from Kristen, but Joji really took over. Kristen went off to start her own lab, and Joji took over the project. Okay, so I'm going to show this model slide, and basically, if we go back to what I just said, we see that when the dams exercise during pregnancy, the offspring has an improvement in glucose tolerance in vivo. And we think that the liver is important, and we can take the liver from the offspring at various ages, and we can start to study the physiology and the epigenetic regulation of the liver. So the first question we wanted to address was, is glucose production changed in the liver of the offspring? Because then this could lead to the improvement in glucose tolerance that we're seeing. So for these studies, and most of the studies I'm going to show you, I just want to remind you that it's a maternal treatment that we're looking at, or is the treatment. And again, sedentary chow, sedentary high-fat trained chow, and trained high-fat fed. And so here's data on basal hepatic glucose production. We've also done clamp studies and looked at glucose production under other conditions, but really remarkable effects here. And you can see that when we look at the hepatocytes, we take the hepatocytes and we do glucose production. You can see if the mother trained, there's an improvement in hepatocyte, a lowering hepatocyte glucose production. If the mom ate a high-fat diet, it's a really elevated hepatic glucose production. But again, if the mom exercised while eating that high-fat diet, the offspring's hepatocytes look extremely healthy with an improved glucose production. So why does glucose production change? Kristen and then Joji looked at hepatic gene expression, and I won't show you the hundreds of pieces of data from that, but what they saw was that there was an improved in gene expression profile of the liver, including genes involved in pyruvate, Krebs cycle activity, and fatty acid metabolism. So one of the questions when Joji started this work, when he came to the lab, is he really wanted to understand at what stage in the offspring development do the changes in hepatic gene expression occur. And so he did studies looking at gene expression from the adult animals down to the weaned neonates and all the way to the blastocysts, which he isolated. And what he found was that these changes in the offspring, in the offspring gene expression occurred as early as day E13.5, which suggests epigenetic changes. So we've had a nice introduction to epigenetics today, so I don't really have to go into any detail here, but just to remind you that these are heritable changes in gene function that do not involve changes in the DNA sequence, things that can regulate, you can measure, you can look at DNA methylation, histone modification, and we actually have a paper that we've just published this past year on histone modifications. I won't have time to talk about that today, and non-coding RNAs. And as we've heard today, if the CPG islands are highly methylated, transcription factors can't bind, but if you have DNA demethylation, you can promote gene transcription. This is a little bit oversimplified, but this is the main idea. So, and we can measure this. We can get an indication of methylation and demethylation. You can look at 5-methylcytosine or 5-MC, and we can also look at hydroxymethylcytosine or 5-HMC, which shows transcriptional activity recovered. We've done detailed studies of both 5-MC and 5-HMC, but I'm going to focus on 5-HMC as it's a little bit easier to show. So what we wanted to understand was, and what we hypothesized was, that if an animal ate a high-fat diet and then we looked at their liver, we asked the question, does maternal high-fat feeding decrease 5-HMC? And if we exercise the moms and look at the offspring liver, does maternal exercise increase 5-HMC? And we can measure global 5-HMC, and we can also look at the 5-HMC levels at promoters of specific liver metabolic genes that we know have been affected by the maternal exercise. So here's some data looking at 5-HMC generation at the promoters of some metabolic genes in the offspring liver. Here's PCX, which is a pyruvate metabolism. And just as we hypothesized, the high-fat diet had an effect to decrease 5-HMC levels, suggesting transcriptional inactivation, whereas with exercise training, you can see if a mom had the chow condition, the offspring liver had an increase in 5-HMC at the promoter of PCX, and that the exercise was able to at least partially reverse the bad effects of the high-fat diet. And when Joji looked at all the different genes, I'm just showing a few here, he saw the same effect, the increase with exercise and the decrease with high-fat feeding. So then, again, as I mentioned, I'm going to show you a lot of cartoons. There is a lot more data in some of our papers. But basically, under high-fat fed conditions, then, we see hypermethylation of the liver, worsened gene expression, and we see decreased glucose production and decreased glucose tolerance. However, with exercise, this is able to be reversed in these offspring livers through and we believe this is through the demethylation. Okay, so we began to see this, and then the next question is, well, what is regulating the demethylation? How is this happening in the offspring liver? And what we knew from the literature was that the regulation of methylation can occur through these 10-11 translocase or TET enzymes, which we also heard about this morning. And so we wanted to determine whether maternal exercise training could regulate TET activity in the offspring liver. What we also knew was that there's a isocitrate dehydrogenase alpha-ketoglutarate mechanism that can regulate TET activity. So we also looked to see whether maternal exercise could regulate these proteins. So here are the data, and now, and this is from the liver. These are from day E13.5 embryos, and you can see, sure enough, that training increases the expression of these TET proteins, TET1 and TET2. We did not see TET3 expression, as well as the IDH, and also an increase in alpha-ketoglutarate activity. So we see that these signaling pathways are up, and so the next question is, what's regulating these proteins in these offspring liver? And Joji said, well, my lab studies AMP kinase and has for years. It must be AMP kinase because exercise always activates AMP kinase. So he looked at AMP kinase, and sure enough, he saw that AMP kinase phosphorylation was significantly increased in response to maternal training in these offspring liver. So basically what Joji found then was that with maternal exercise, when we look at the offspring liver, you get an activation of AMPK, you get an activation of this signaling mechanism that then can regulate the demethylation and epigenetic changes. Now I haven't shown you all the data, but he's done knockdown experiments, overexpression experiments to show that this pathway in the liver is regulating in this means. And that then these changes then result in an improved gene expression, leading to this decrease in glucose production and the improved glucose tolerance. So maternal exercise regulates this offspring liver metabolism via the activation of this AMPK TET signaling mechanism. So the next question then was, how does the exercising mother stimulate this AMPK TET signaling, leading to these changes? And this now leads to this part of the talk on where identification of a placental protein that we believe is mediating improved offspring health. So what Joji did to try to get to this was, I think he did a very clever experiment where he took the serum from a pregnant female that was sedentary, and he also took serum from a female that was pregnant and had been trained through the voluntary wheel caging. But he also took serum from a trained female that was not pregnant. And he did a serum stimulation experiment, so he isolated primary hepatoblasts just from animals that had not been treated, their mothers had not been treated, and he did this serum stimulation for 24 hours, and then he looked at the analysis of the AMPK, this AMPK TET signaling axis. So here on the left, I'm just reminding you of this AMPK axis that we saw was activated by maternal exercise. So if we look at the TET1 protein shown here, this is an expression of TET1. What we see is then when he stimulates with serum from a sedentary female who's pregnant, you see no significant effect on TET1. But if you take the serum from the mom that was trained, and you see an increase in TET1. But when he took the serum from a female that was trained but not pregnant, this effect of training was completely gone. So he only saw the increase in TET1 protein when the serum was from a female that was trained and pregnant. And when he looked at all the other proteins in the TET and IDH signaling pathway, he saw the same thing, as well as AMP kinase shown here. No effect if the female was trained but not pregnant. And when he looked at the various genes, and he also did methylation and demethylation, he saw the same thing. So the entire signaling pathway was only upregulated if the serum was from a trained pregnant female. So this then led us to the question is, well, what is in the serum from the trained dams that's responsible for the activation of this signaling axis? So to do this, he collaborated with Harvard lab to do mass spec on serum. And here he did the serum from the sedentary dam and also a trained dam. And so when they did the mass spec, they found 577 proteins. So that was nice, a lot of proteins, but we certainly have to narrow it down to look for a factor or factors important in the serum. So what he did next was Joji took the data, and he only took proteins that had been increased by at least 1.5-fold in serum from the trained dams. And he did increase because he figured that we're going to start with proteins that are increased, and he had this cutoff of 1.5. So this narrowed it down to 78. Next, he reasoned that this must be a secreted protein if it's going to have an effect on in the fetal tissues. And so 19 proteins had been categorized as secreted proteins. Of these, 4 proteins were known to be expressed in placenta. And remember that it has to be, this protein needs to be coming from the placenta because the effect is only present when the female is both trained and pregnant. And then he thought about AMP kinase and that the AMP kinase was stimulated, and it turned out there was just one of these proteins that had been previously reported to phosphorylate AMP kinase. So what is this protein? So the protein is superoxide dismutase 3, or SOD3, also known as extracellular SOD or EC-SOD. It has a classical role as an antioxidant protein, but more recently it's also been shown to have roles as signaling proteins, including activation of AMPK, as I mentioned. So does SOD3 actually change? And sure enough, it does. So if you look at the serum of the trained and pregnant dams, you can see that training increases serum SOD3, and that this occurs under both chow and high-fat conditions of the mother. So again, this is in the mothers, the serum SOD3. if we then look at the serum of the females that were not pregnant, there was no effect there was no effect of training to increase SOD3. So again, likely coming from the placenta. And when then Joji looked at the mRNA expression in all the different tissues from the trained dams, including the placenta and the trophoblasts of the placenta, what you can see over here on the right is that the training increases SOD3 in the placenta as well as the trophoblasts of the placenta, but does not increase in any of the other tissues in the mothers. So we think that SOD3 is activating this signaling pathway, and so we wanted to prove, though, that SOD3 was actually important and able to activate this. So the next question was, could we just take recombinant SOD3, and could this mimic the effects of exercise to induce these epigenetic changes in the offspring liver? And so again, he used primary hepatoblasts from untreated mothers, and he treated these cells with SOD3. And again, here's the signaling pathway that we're looking at, and sure enough, recombinant SOD3 throughout the time course can increase AMPK phosphorylation, can also increase the expression of the proteins, of the TET and the IDH proteins, as well as increase the expression of some of these key genes, key metabolic genes in the liver. Again, Joji did all the knockdown experiments, et cetera, to show the specificity of this. So then we have a situation that we believe where when an animal trains, a pregnant female trains, you get an increase in SOD3 in the placenta, that this then goes through the maternal artery, so you can see the increase in the serum and also in the placenta, and then the SOD3 then can go to the fetal liver where it has its effects. And this is the signaling effects then that we see within the liver. So our hypothesis then was that placental-derived SOD3 could mediate the benefits of maternal exercise on offspring health. But to really understand whether this was an in vivo effect, what we needed to do then was to generate a placental-specific SOD3 knockdown, and this is what Joji did. The kind of the beauty of this type of model is when you're studying these embryos is that you have both the wild type and knockout are in the same mother, so they have the same treatment. So it's a nicely controlled experiment, and then you can either have a sedentary mom or a trained mom. And this worked very nicely, knocking down this specifically in the placenta. Here you can see in the floxed animals the increase in SOD3 with training that we see, and then this is knocked down in the knockout, and there is no effective training, and this is quantitation of this data over here. So we have a really good model to study SOD3 in the placenta. And now I'm going to show you 5-HMC data in these knockouts, which are shown in green on the right-hand side. Here's global 5-HMC. Remember, when this goes up, it suggests transcriptional activation. Here's in the floxed animals. Again, we see this effect of maternal exercise to increase global 5-HMC, and a significant decrease even in the sedentary animals if there's no SOD3 in the placenta and no training effect. When we looked at 5-HMC at the promoters of various genes, we see the same thing. Here's the PCX gene, which I showed you before. Here's the increase with training. But if there was no SOD3 in the placenta, lower 5-HMC and no effective training. Same thing with these other genes. So these data really show that if you knock out SOD3 in the placenta, that you get a deactivation of this whole signaling pathway. Again, there's much more data to show all the different aspects of this. But the other thing that we really wanted to understand is that we see knockdown of the signaling pathway. Does this translate into changes in glucose tolerance in the offspring as the animals age? So we measured glucose tolerance and serum insulin concentrations in offspring up until age 52 weeks of age. And here are data from the glucose tolerance test. And you can see, again, in the flocked animals, as we had seen before, improved glucose tolerance if the mom had trained. But in the offspring, where they didn't have SOD3 in their placenta, we see a worsening of glucose tolerance even in the basal state, and no improvement in glucose tolerance if these animals trained, if the moms had trained. If we look at insulin, we see the same thing. Insulin is lower if the mom's trained in the flocked animals, but with the knockout, you have an elevation in the liver and no effect of training. So these data really point to SOD3 as mediating the beneficial effects of maternal exercise on offspring health. So we have this model again that I showed you before, and the next question then is, how does exercise lead to an increase in placental SOD3? This is really important because if we start thinking clinically, SOD3 may be a mechanism where we can improve the health of the fetus and potentially the baby. So we really want to go further into this. So, I don't need to explain this concept here that during exercise you have secretion of proteins from muscle, adipose, heart, other tissues that are exercise transducing molecules. And so certainly this can happen in a pregnant animal that's exercising too. So we reasoned that there was these factors that would then affect transcription factors that then in turn could be transcription factors to regulate SOD3. So what Joji did then was he did an RNA-seq experiment and took the placenta from sedentary animals and trained animals and did the RNA-seq. And he found 26 different transcription factors that were significantly regulated. And one that was of particular interest to us was VDR, or the vitamin D receptor. It was highly elevated. And when he did experiments, when he did luciferase assays, he showed indeed VDR could be a transcription factor for SOD3. But here you can see that if you look at the trophoblasts of the placenta, in trained animals, the VDR, the vitamin D receptor, was increased, whereas cofactors were not. And so we wanted then so we started to think, okay, so vitamin D must be very important in this effect of exercise to promote the metabolic health of the offspring. So we wanted to ask the question, is vitamin D necessary for these effects of maternal exercise to improve offspring health? And we could do this by feeding the animals a low vitamin D diet. And we could also ask the question, can vitamin D treatment function as an exercise mimetic? So can you just take vitamin D and get these beneficial effects of exercise to improve the offspring metabolic health? And we can do that by feeding the animals a high vitamin D diet. So there we go. So again, this is the typical paradigm that we use with sedentary and trained moms. But in this experiment, the moms were either fed a normal vitamin D diet, a low vitamin D diet, or a high vitamin D diet. Again, the breeders were just normal. They were fed a normal vitamin D diet, and the offspring were also normal. So we looked at levels of SOD3 and saw the expected change, but what I want to show you, the key point is what happens when the moms are eating these different vitamin D diets and what's the effect on offspring glucose tolerance. So here, this is the normal vitamin D diet, and you can see again, we see this improvement in glucose tolerance if the mom was trained. This is the offspring glucose tolerance again. But if the mom ate a low vitamin D diet, there's a little bit of a worsening of the glucose tolerance, and also there is no effect whatsoever of the mom's exercise on the offspring health. Now, when the mom ate a high vitamin D diet, if you compare the green bar with the yellow bar, it's actually not significantly, statistically significant. So there's no effect of just eating the high vitamin D diet, but there is a good effect with the combination of the training and the high vitamin D. So then to summarize this, what we see is that normal vitamin D intake is necessary for maternal exercise to improve offspring metabolic health, and vitamin D treatment alone does not function as an exercise mimetic to improve offspring metabolic health. And we've done other experiments in the SOD3 knockout animals to show that you really need placental-derived SOD3 and vitamin D in the diet to mediate the beneficial effects of maternal exercise on offspring metabolic health. Okay, so this is all animal work. Of course, our goal is to see if we can translate this. So one of the things that we wanted to do was to understand whether exercise would actually change SOD3 in pregnant women. So in collaboration with a group from Ottawa, Celine Agur and Christy Adamo, we did experiments where we compared women that were either inactive, they were very inactive with 2 minutes per day of moderate physical activity, or were highly active, about 40 minutes per day. And so what we did was we looked at SOD3 in the serum of these mothers. This is in the blood collected in the second trimester, and you can see, sure enough, SOD3 is significantly increased in these women. And when they collected the placenta at birth, we looked at the placenta SOD3, and this was also significantly increased in the women that were more highly active, and this correlated very closely with the amount of physical activity that they did. So this was really exciting, we're really looking forward to continuing to work on the human studies. But just to summarize this is from Joji's paper. This is the cartoon that gives a lot of detail of what I've talked about. But we see that SOD3 is increased in serum and placenta for physically active pregnant mice and women, that maternal exercise activated VDR signaling is necessary for placental SOD3 expression, and that placental SOD3 and vitamin D are required for the benefits of maternal exercise on offspring health. So where do we go from here in this part of my talk? One of the things that we think, going back to the animal models, is really important, and a question I get all the time is how much exercise is necessary to have these beneficial effects, and what mode of exercise is needed, and can we use some drugs to mimic the effects of exercise? So we have ongoing studies now looking at different intensity of exercise, different modes of exercise, and different possibilities to see if we can mimic these effects of exercise. We also want to study the placenta more. We see a role for VDR and SOD3, but we know there are a lot of other proteins that are changing in the placenta, and so we're doing work on the placental secretome and really trying to understand if there are other factors that are coming out of the placenta when the mom exercises. We also want to try to understand what are the signals from the mouse, from the mother to the placenta, and we have some ideas there that there may be specific tissue-specific effects that are then leading to changes in the placenta. And then finally, we really have focused primarily on the liver, but we think there are other tissues that may be really important in modifying the health of the offspring that are being affected by all these processes, including the adipose tissue, brown fat, obviously muscle, and the brain. And then in terms of human studies, there's lots we would like to continue to do. We're working now on the role of vitamin D in the pregnant women, the optimal, and similar to the animal work, what are the optimal maternal exercise paradigms, and how does maternal exercise alter the placenta? So in the last couple minutes, I'm just going to show you very briefly this work that we just published and this is work done by Anna Alves Wagner from the laboratory. And her hypothesis was that physical activity of the grandmothers will improve the metabolic phenotype of the F2 offspring. And so these are really very difficult studies to do, heroic, as one of the reviewers of our paper actually called them, which I've never seen that before. And it's because so many, it takes years to do these studies, even using a mouse model. But basically, Anna treated the F0 as we had before for all the studies on the first generation. And then she took the F1 generation, and when the F1 pups from each litter are 8 weeks of old, one F1 mouse with an average body weight was selected to breed with untreated females. Now, I'm going to show you data from F1 males. We're also doing the studies in F1 females. And then she breeds them, and then she looks at the F2, or the grand offspring, and this is studied out again to a year of age. And again, the treatment is only of the grandmothers. There's no treatment of the F1 or the F2. So grand maternal exercise didn't have effects on F2 in terms of litter size, male to female ratio, food intake, or exercise capacity. But what we really were interested in looking at, again, was the glucose tolerance and the insulin concentrations. And so here are the data. These are the sedentary in the black, and if the grand maternal exercise, the grand offspring from the grand maternal exercise in blue. And again, as we've seen before, these are data from male offspring. As the animal ages, there's a worsening of glucose tolerance. But what Anna found, quite remarkably, was that if the grandmother had exercised, the grand offspring had lower area under the curve, improved glucose tolerance compared to offspring where the grandmother had been sedentary. So really striking findings. And again, there was no difference in the food intake or activity levels of the offspring. This is the female offspring, and we see the same thing. Interestingly, the females become much more glucose intolerant as they age. But again, if the grandmothers had exercised, the grand offspring, glucose tolerance was improved. Some other interesting things is that if we look at the males and we look at insulin or HOMA-IR, if the grandmothers had exercised, the insulins and HOMA-IR were lower, which is good. It's also interesting, if you put males and females side by side, you can see even at baseline, females' insulin concentrations at 50 weeks are much lower, and there wasn't a significant improvement in this, but we think this may be because they're already very low. If we look at some mass parameters, we see, in the males, we see a significant increase in lean mass, a significant decrease in fat mass, and interestingly, higher bone mineral density if the grandmother had exercised. These effects were not really prevalent so much in the females, but in the male offspring. So to summarize then, grand maternal exercise improves the metabolic health of male and female F2 offspring. Where do we go here? Well, again, there's a lot to do. We want to try to understand the mechanism of this. We want to look at this. We are looking at the sperm and the oocytes for epigenetic regulation and small RNA analysis. We're also doing studies in both the, as I said, both in the F1s from males and females. And we want to understand more about what's happening in the F2s, looking at their RER, food intake, activity levels, and also looking at multiple tissues from the F2s to understand what's happening there. So the title of the talk was, Should Women Exercise During Pregnancy? Well, I am not an obstetrician, so I can't say that, but what I can say is that the data from the animals suggests that maternal exercise may be a means to reduce the transmission of increased risks of metabolic disease to offspring. We want to do more human studies. There are a lot of human studies out there. Most of them are looking at the mother or the child when their children are young. But I think that the data are very clear that if you can, when a woman is pregnant, increased physical activity or have high levels of physical activity, or at least some physical activity, is probably very important to the health of your offspring. And we feel that determining the physiological and epigenetic mechanisms that mediate these benefits of maternal exercise on offspring health can potentially lead to new therapies to prevent this transmission of metabolic disease. Because if we can translate this work to humans, it could have really enormous implications for the prevention of obesity and type 2 diabetes through multiple generations. So with that, I'll stop. I highlighted the work of Joji and Anna and Kristen, who've just done amazing research over the years. Many other people involved in this work. These are former and current members of the lab. Mike Hirschman, our lab manager, involved in everything and so critical. Mary Elizabeth Patty at Joslin. Our collaborators from Ottawa, which has been great. And then we have our collaborators in Colorado working on the SOD3 with us, Eva Grashik supplying animals, and also collaborators in Denmark who are working on studies right now, human studies right now with us as well. And finally, I want to acknowledge the NIDDK, which has funded this research that I've talked about today, and a lot of the fellows have had support from various other additional organizations that is also greatly appreciated. So with that, I'll stop, and I don't know if there's questions, but I'm happy to talk. So.
I'm sure there's questions. Totally agree with that reviewer on the heroic adjective there. That is fantastic.
Yeah.
Okay.
Ren Yan from University of Virginia. Beautiful work, as always. I tried to understand the link from ECSOD to AMPK activation. So it's a superoxide dismutase.
Yeah.
Generally speaking, it's outside, bind, maybe internalized into the cell. And how does the superoxide dismutase activate AMPK phosphorylation?
Yeah, so that's something that we've definitely looked at a little bit. We've looked at H2O2 to see if that's a potential mechanism. That doesn't seem to be the case. At this point, we don't know. We don't know. We don't see really changes in LKB1, so we don't really know the mechanism for the AMPK activation. So it's something that we're working on.
The second one is that here, obviously, that ECSOD is not a ligand for receptor, so it's likely to get into the cell.
Right.
There is a mutant called R213G, which is a human SNP that cannot bind to the extracellular epitope that cannot be internalized. That might be a mouse that would be test out.
Yeah,
we have
that's
those
a really
mice.
good experiment. Yeah, that would be a great experiment.
Thank you.
Thank you.
Okay, let's go over here, and then we'll go over there. Okay?
Hi, I'm Yun Tian. I'm from UVA, from Zen's lab. So I actually want to ask the same question that he asked, but I want to add maybe two sub-questions to that. So in your the mass spec from the, I think it was either the serum or placenta where you found the SOD3 that was increased, do you see other antioxidant that's also increased, maybe in the, in the 79 other proteins that you found in there?
We saw about 150 different proteins changed, and I don't recall that we saw other antioxidants. So, so you're talking about with the okay, yes. So there were about 80 different proteins. So I don't recall that we saw other antioxidants, but that's a good point. We can go back and we can look at that to see if there are other antioxidants.
Yeah, the reason I asked that question is because I was curious whether the SOD3 is regulatory to AMPK or is it also being antioxidant, because also vitamin D.
So we have done experiments where we've treated with NAC, and we don't see an effect when we do that. So, I mean, it may be specifics for SOD3, but that doesn't rule out there couldn't be other antioxidants that are functioning in that manner.
Yeah, and the second point is when you did you look at maybe the oxidative stress in the placenta or in the embryo, and still the same point with the
yeah, we're starting to do those types of experiments looking at ROS and see, and we think that could contribute to some of the effects, but we don't know for sure at this point.
Okay, thank you.
Yeah, hi, Matea, York University, great presentation. My question, if I understood correctly, you mentioned that SOD3 goes up pretty early in the developmental stage, like day E13.5 you mentioned.
Yeah.
But we only see improvement in glucose tolerance pretty late, like at 52 weeks, I think you mentioned.
Right.
So how do you reconcile that delay in between those two?
Yeah, so I think that We see the effects on gene expression and methylation very early on, as early as day E13.5, SOD3 and the whole pathway. We don't see the effects, depending on the measurement we're doing, we may see something as early as 24 weeks, but certainly nothing earlier than that. And I would hypothesize that the system is there and it's in place, but there really, in younger ages, there isn't any glucose intolerance. There's, the animals are fairly healthy, insulin. So the effects don't really seem to appear until the animal becomes less healthy. And so that's what I believe is the reason why we don't see effects until later on.
Awesome, thank you.
Thank you.
Okay, hi Laurie.
Hi, Marnie.
Fascinating talk, really.
Thank you.
So SOD3 is a really interesting protein. It can be released by vascular and vascular-associated cells.
Right.
I was just curious if you could comment on the SOD3 knockout model, and perhaps also comment on whether vascular cells, which are enriched in the placenta, might also contribute to the SOD3.
Yeah, so the experiments that we did with the SOD3 were specific for the placenta. So it was a trophoblast-specific knockout. So our model doesn't really look at the global effects of knocking out SOD3. We do think that SOD3 probably does have a broader role in the development of the fetus. That's one of the things that we really want to do is start to look at other tissues and how SOD3 may be regulating brain, or vasculature is a very important area, as you mentioned, and really look at all the different tissues to see how SOD3 may be affecting these tissues. So,
thank you.
Go ahead.
Hi, great presentation. I just have a question. Do you know if there have been any studies examining the male's activity level before, like, going into conception of the babies?
yeah.
Or what effect that might have?
Yeah, so we've actually have a whole other line of research that's related to this, where we've done studies where the males have been in wheel cages, are housed in wheel cages prior to conception. We see that there is an improvement in glucose tolerance in the offspring if the males are exercised. Actually, this was Kristen Stanford's paper that she published. And we also see changes in the microRNAs in the sperm that we believe is part of the mechanism. We haven't figured out the whole mechanism, but definitely paternal exercise also has important effects on the offspring metabolic health and can improve the offspring metabolic health.
Great, thank you.
So one more question.
It was very fascinating, the data, and it's Rolando Sedia from York University. I was curious to see, when you did the vitamin D experiments, you used low and consider normal and
Low, normal, and high, yeah.
So what is a normal vitamin D concentration in a mouse? Because those seem to be relevant to humans. And also when you do the feeding, how much higher or how much lower does it go compared to an animal that is fed a normal diet under normal conditions?
Yeah, so the normal vitamin D is, the commercial chow that you get that gives the normal levels. And I'm trying to remember off the top of my head what the normal levels are, around 100. But I know that the difference between the low and the high is about 10-fold difference. But it's actually on my slide, the actual values, but I can't remember them off the top of my head. But it's about a 10-fold difference between the low and the high.
But did it increase tenfold when you did like 10,000
Yeah, so we measured the
units
vitamin D levels to make sure that the diet actually changed the levels, and they did.
Thank you.
Great. Thank you very much for a great presentation.